Welcome to today's CanPath webinar. My name is John McLaughlin. I'm a professor emeritus at the Dalla Lana School of Public Health here at the University of Toronto, where I previously served with the CanPath team as executive director. And it's my great pleasure to be back with you today for this webinar. Thank you all for joining us. And I'll get started with the land acknowledgement. So we acknowledge the traditional territories of the Mississauga of the New Credit First Nations, Anishinaabe, Wendat, Huron, and Haudenosaunee Indigenous people on which CanPath's National Coordinating Centre at the University of Toronto now stands. The territory was subject to the Dish with One Spoon Wampum Covenant, an agreement between the Iroquois Confederacy and the Confederacy of the Ojibwe and Allied Nations to peacefully share and care for the resources around the Great Lakes. With CanPath teams working across Canada from coast to coast, we also wish to acknowledge the ancestral territories that are the home to many Indigenous people from across Turtle Island, and we are grateful for the opportunity to work with you on this land. Two presenters today, Dr. Philip Awadalla, Professor of Molecular Genetics at the University of Oxford and at the University of Toronto in both molecular biology and public health. He also holds leadership roles at the Ontario Institute for Cancer Research, among many other appointments. And he is the National Scientific Co-Director of CanPath. Philip will show some of how Ontario's genomic and genotyping data have been utilized in a wide range of genomic studies. Dr. Guillaume Lettre, Professor of Medicine at the Montreal Heart Institute and the University of Montreal, and also serving as scientific co-director of CARTaGENE in Quebec. Guillaume will present on genetic ancestry in Quebec, French-Canadian founder effects, and findings from sequencing studies of the CARTaGENE participants. So I thank both of you as tremendous leaders of science for Canada, but for putting such good use to the CanPath and provincial regional cohort data systems and resources. Very briefly on housekeeping, after the presentations, there will be time for questions. Please use the Q&A button at the bottom of the Zoom window to drop your questions through the talk, and we'll get to them soon. So with no further ado, let's hear from Dr. Awadalla first.
Great. Thank you very much, John, for that introduction. It's great to have you and see you again here. And I'm very pleased as well to be co-presenting today with Guillaume, who's helping co-lead the CARTaGENE activity as well. So in my presentation today, I'm hoping to largely focus on activities we're doing with the genotyping and the genomics that we've been collecting in the Ontario cohort. But what I thought I'd first do in this presentation is to remind people of CanPath, reintroduce people to the CanPath activity, and then I'll be talking to some extent about work we're doing with some of the genotyping data from both Ontario and Quebec, as well as data that we're collecting at the single cell level from Ontario, and also work we're doing developing liquid biopsies using material, both biologics as well as data captured from Ontario Health Study participants as well. Getting into this, I still lead a lab in Ontario, but also growing a lab at Oxford as well. And so much of the material I'll be talking about today has been developed by the first three students, including a former student, Kimberly Skeed, Nicholas Cheng, and Jasmine Kang, who've been working with me on the Ontario Health Study genomics activities. So as I said, the first thing we're going to be talking about today is CanPath and why do we have a population cohort in Canada. So we like to make the case that population studies where we recruit participants, both healthy as well as unhealthy, or people who have been diagnosed with a disease, are incredibly important for identifying the underlying determinants of disease and disease development and understanding how these diseases develop over time. And so we've been using CanPath for much of our research in my lab. And this is the makeup of CanPath. We've recruited over 360,000 participants. You can see Ontario and Quebec there in terms of our numbers. Sorry that CARTaGENE got caught off with the logo, but 43,609 participants were recruited in CARTaGENE. And in Ontario, there are over 181,000 participants of the OHS study that are part of the CanPath activity. From all the CanPath participants, they've consented to be part of to allow for generally broad health research. Participants complete a questionnaire. They've consented to allow us to follow up their health through both questionnaires and through linkages through administrative studies or linkages to administrative data. And for many of our participants, and of course, this is where the genomics comes from, we've collected biologics from many of these participants as well. This is the CanPath leadership, and as John had mentioned during the introduction, I co-lead the CanPath activity with Jen Brooks. Missing from this slide is Vicky Kirsch, who's just taken on the leadership of the Ontario Health Study. And of course, we've got the full CanPath team here and all the leaders of the regional activities here on this slide as well. It's a great team to work with. As I mentioned, we capture information from participants, both healthy and from people who have been diagnosed with diseases. Some of this is self-reported, and so some of this information that you're seeing on this slide is the frequencies of those outcomes as self-reported by our participants at baseline. This is a longitudinal cohort, of course, so our participants are, of course, developing conditions over time, and so these numbers, of course, are the prevalent conditions at baseline, but this prospective look at health in Canada is obviously a very important component of our activity, and we have, through our consent, the ability to contact our participants and look prospectively for the next at least 20 or 30 years. Now, I like to throw this slide out there as an important slide that John Bell, who's the Regis Professor at Oxford, once asked me, have we solved the Canadian problem? And the Canadian problem is our ability to do these types of linkages to administrative health data, both at the provincial and as well as at the national level. One of the activities that's ongoing is our partnership with the Health Data Research Network in Canada. It's a support unit funded activity where effectively through a federated way of access to linkages in the various provinces, researchers can get access to both CanPath participant information as well as administrative health data in each of the hosted in each of the provinces. But fairly recently, we've also signed an important collaboration with the Canadian Institute for Health Information, where CanPath as a national activity can link to this national administrative health resource as well. And that's one mechanism by which a researcher like yourself could get access both to CanPath data as well as linked outcome data at a national level as well. And this was just signed not that long ago. I think it was in September or October. And of course, that kind of information, as I mentioned before, it allows us to capture both prevalent data, and so I'm showing some numbers here for prevalent cancers, as well as, say, incident cancers. Those are that's information captured through those linkages about, say, in this context, individuals who have, say, developed a cancer since they became part of the cohort as well. Okay, so I'm going to get into now some of how that's an overview of CanPath, and just really quickly, Guillaume and I are going to be talking about some of the genotyping and genomics that's been captured in at least two of the regions, Ontario and Quebec. This is just a principal component plot of data captured from some of that genotyping data. You can see here on the right that 20,000 participants have thus far been genotyped with arrays in the Ontario Health Study, and around 30,000, and Guillaume's going to spend more time talking about what's been done in Quebec. And I don't want to ignore that there has been genotyping work captured in the other regions, some of this through CanPath and some through other activities as well. I'm just highlighting one. Here's one called the Confluence Project, which is a major breast cancer GWAS initiative where CanPath participants CanPath data samples have been contributed to this genotyping activity. And very recently, this was just announced last week actually, our partners in Manitoba and Saskatchewan were awarded funding through through Genome Canada to have genomes captured from the prairie provinces as well. So we're quite excited and happy for our team members there. Okay, so let's get into some science now. So that's an overview of CanPath, and I'm going to talk about now what we've been trying to do with genotyping as well as single-cell omics in the Ontario Health Study. And then I'm going to lead into talking very quickly, hopefully, about developments in the liquid nanopore space before turning it over to Guillaume. So as I mentioned before, CanPath has done extensive amount of genotyping in 2 provinces thus far, Ontario and Quebec. And we've also got access in some of the work that I'm about to show you today to material captured from a partnership and collaboration called the H3 Africa Project. And the countries on the right are some of the countries where we've captured some genotyping data as well. And so one of the projects that we've been interested in using this genotyping data for isn't so much to look at, say, how germline variants contribute to health and how we age, but it's actually how we can use some of the genotyping data that we currently have to understand somatic evolution, or how somatic mutations accumulate with time. In some ways, that somatic mutation burden accumulating in our blood is associated with, say, a decline in health as we age. But in some participants, some of those mutations or how we age could be considered a healthy aging process. And we're quite interested in both. We're interested in both how somatic mutations might contribute to aging, both from a decline in fitness, and also identifying individuals in our large cohorts who actually we might consider or call healthy agers. So just a quick overview. So for those who haven't heard about this phenomenon, one particular phenomenon where somatic mutations accumulate in blood and where those mutations might give a fitness advantage to, say, those blood cells or those blood stem cells where those mutations occur is a phenomenon called clonal hematopoiesis. And that's an idea where somatic mutations might occur in the blood stem cell. And then if that mutation takes over, then your stem cells in your blood, effectively almost all might have that mutation, or many of your stem cells will have those mutations. And so that might be driven through a positive selection kind of process. Those mutations might also just, accumulate through genetic drift, but also mutations might, like that, also probably, many of them are probably removed from your blood as well. And this has now become quite a field of research now, particularly in the hematological space and in the cancer as well as cardiovascular space, where we now know that there are a number of driver mutations where these point mutations occur at, say, some of these genes that I have on the screen here, and these mutations can start to take over. And by the time you're about 80 or 85 years old, and many of us, these mutations are extremely prevalent. Some of these mutations are point mutations, but some mutations are chromosomal or larger scale alterations. And I'm highlighting these chromosomal alterations here because we know in big population cohorts like the UK Biobank, as well as now in CanPath, that these things accumulate in our blood as we age and probably have an impact on both our fitness, molecular phenotypes, and potentially a whole broad range of phenotypes as well. So what we've been doing in CanPath is looking at how these mutations might accumulate. And we've been looking at this in our CanPath cohort to try to identify how these somatic mutations might accumulate, not just in CanPath, but also how we compare to regions in, say, Africa as well. Do we accumulate, say, hotspots of these mutations in our genomes that are distinct than in Europeans or people of European ancestry, or people with different environmental exposures or infectious exposures compared to individuals who might be exposed to different environments or infectious diseases like what we see in Africa as well. So this is just a quick snapshot of the genetic diversity we see in Africa, for example, as well. And so the populations you see in Africa span both north, south, east, and west. And what we've been doing with CanPath is mapping hotspots of chromosomal alterations in our Canadian cohorts or North American cohorts, and that's what's highlighted here in yellow in our 22 chromosomes here. And this is data coming from both the Ontario, this is actually data from coming from both, yes, the Ontario Health Study and the CARTaGENE study. And in blue here are some hotspots, and we're highlighting here, say, chromosome 9, where there seems to be neighboring hotspots occurring in slightly different regions in North America versus Africa as well. So this is a pretty exciting study for us. And and here's just kind of a breakdown of some of these, how these things map in a circus plot. And you can see here that there are places in, say, North America that we don't see hotspots, and there are where we see, say, a gain of function mutation or gain, sorry, of a chromosomal alteration in Africa as well. So, and then of course, just focusing on in North America here, some of these chromosomal alterations, we're doing a zoom out here on chromosome 2, actually seem to overlap some of the known drivers of clonal hematopoiesis, some of which are known to be indicators or drive things like leukemia and other blood cancers as well. So, oh yeah, and yet just to that point as well, sorry, and here's just looking at some data that's showing us that the frequencies of these somatic mutations also has a significant impact on risk for cancers as well, and that's what these hazard ratios are showing us there on the right, that the number of mosaic chromosomal alterations captured with these genotyping data is associated with risk of cancer in the CanPath cohort. I just want to highlight some work now that we're about to do, and this is data that I'm hoping will be completed in terms of collection. We're now going to look at how these chromosomal alterations in a longitudinal way change, because we do have longitudinal sampling in Ontario, and we're going to be looking at how chromosomal alterations accumulate with time. And at the same time, we now have a program with Olink, and we're collecting this data now where we're looking at proteomic data at the same time points and from the same samples. And we want to investigate how chromosomal alterations will impact these molecular phenotypes, both in people who have already had a chromosomal alteration in their blood at baseline, as well as those individuals who didn't, and then might have accumulated a chromosomal alteration with time as well. So this is work that's ongoing presently. Okay, now that's how I wanted to describe what we're doing with chromosomal alterations, with genotyping data. Some other things that we're doing now in the Ontario Health Study is not just looking at how chromosomal alterations or mutations impact your health and decline in fitness, but as I mentioned before, we're also interested in those people in our Ontario Health Study who are healthy agers, right? Who have the blood phenotypes that are of an 80-year-old who actually, but that could look like a 50-year-old in terms of their blood traits or phenotypes. So we've been working specifically on identifying individuals with just that type of phenotype using, say, complete blood cell information that we have. And we've developed something that's kind of a modification of what's called the Intermountain Risk Score, where we use our cell count information that we have on all the participants who've provided blood samples, use those CBC information to kind of characterize our individuals in our cohort and identify, say, who are those healthy 80-year-olds versus unhealthy 80-year-olds, or who are those 80-year-olds who have a blood phenotype, say, maybe of a 50-year-old. And this was work that was started by my student Alyssa Bader. It's now a work that my newer student June Kim is also now working on, where we have in this figure here the CRS is our complete blood count risk score. We stratified in this picture people by age, and this allows us to identify people who are aged. That's what the A stands for, and L stands for low risk, and versus H, which is high risk, and young people who are low risk versus young people that are high risk. And we can make all kinds of comparisons, and looking at their blood phenotypes, we can look at, say, what makes a low-risk aged individual different than a high-risk aged individual. And actually, one of the things that I found quite interesting just from looking at complete blood counts is that RDWCV, which is the coefficient of variation of red blood cell width, is actually a really good indicator of healthy aging blood or high risk, low risk blood as well. And this is just these are just kind of the variables you captured with CBCs. So we've been taking about 100 participants from each of those 4 quadrants that I just shown you, and with the ACD tubes that we have, we've been able to capture single cell transcriptomics from those participants because we're interested in that question at the single cell level. What are the factors both in terms of cell types as well as genes expressed within those cell types that might be driving healthy aging as well? This is just a map of one of our first maps, in fact, of what that data looks like at the single cell level. So this is just a highlight for you, the community, that with our material that we've stored and cryopreserved is suitable for single cell assays. And what we've been able to capture using, say, differential gene expression at single cell type resolution are sex and cell type specific markers that seem to be able to discriminate healthy versus unhealthy or high risk versus low risk blood types as well. What we've got on the different axes here are male and female samples, information coming from males and females. And you can see here that many of these factors are sex specific. They're either on the line right on the axes so that they don't actually discriminate very much within the other sex, but they do within one sex. And there are a few factors that seem to be found between both sexes as well. And just to highlight some of the findings here, and this is a paper that's now in review, is that much of the gene expression data or differential gene expression that explains the variation between a high risk and low risk individual and aged individuals are largely features that are common in an older individual who's low risk, and we see that they have some of the same gene expression features as a younger individual. And that may not be that surprising, but we actually are able to capture other things with all the omic and genotyping data and sequencing data that we have. We're actually doing EQTL mapping to look at how differential gene expression maps at the genomic level. Okay, the last thing I want to profile about work that we're doing in the Ontario Health Study before I turn this over to Guillaume is work that my student Nick Cheng, who's having his Viva next week, next Thursday, and this is in the liquid biopsy space. And in this liquid biopsy space, again, we've been able to, I'm going to use this data and material that we've captured with both incident blood, well, largely with incident blood samples. And in the case of incident blood samples, we have blood captured before somebody has diagnosed it with a cancer. And because we can link through administrative health outcomes who then has gone on to develop a cancer, we can then ask in those plasma samples that we've cryopreserved whether there's a signature of a cancer, a solid tissue cancer, in our data. Now, naturally, everybody knows, and sorry about this slide has been slightly corrupted here, but in any case, we all know that capturing and diagnosing cancer early is certainly much more is associated with higher outcomes or higher survival probabilities associated with treatment. Sorry. And right now, what we've got are a number of, classical, if you like, approaches to screening populations. Mammography for breast cancers, which I'm going to be talking about mostly here, is the way to do it. Now, it's just screen population for early detection, sorry. I think everybody's heard about where the world is going in terms of liquid biopsies. Liquid biopsies is a non-invasive approach to using blood or plasma to potentially detect the signature of where there might be damage somewhere in the body, and it's releasing DNA into the bloodstream. We've been taking a DNA methylation approach like others, and the reason we do that is because DNA methylation is a very tissue-specific marker of where damage might be coming from. So we've been developing tools which, training with, say, things like the TCGA, where it has a big database of DNA methylation, and we can use, say, some of that information and our sequencing approaches to see if we can detect a breast cancer and a prostate cancer early. We've been doing this mostly in the Ontario Health Study, entirely in the Ontario Health Study, except in those places where we've done some validation work, where we've gone in, identified an individual that came into the cohort without a diagnosis, and then followed up. And through linkages to Cancer Care Ontario, we can see whether they developed a breast cancer and a prostate cancer. And you can see here, these are the numbers that are just normalized in terms of time. This is when the blood sample came in at times A0, and then some people have come in and were diagnosed, sorry, with a cancer maybe in a couple of months or say 7 years later. And this is just some of the data that we're looking at here. And in terms of their time sent to diagnosis, the stage where they may have been diagnosed in terms of breast cancer or prostate cancer, and of course pancreatic cancer as well, but I'm going to be mostly showing you breast and prostate work. And as I said before, we've decided to take a DNA methylation approach. And we can see here that when we look at differential methylation using this approach, we can discriminate pretty nicely on the right, the different kinds of cancers that we could potentially be capturing. And when we compare, say, cases to controls, it separates fairly nicely as well in the context of DNA methylation. So you can see here that in color, these are their cancers for breast and prostate and pancreatic, and they discriminate. And the nice thing about a cohort like the Ontario Health Studies, we have a lot of material where we can do a nice design with regard to matched controls for not just sex, but of course, risk factors like drinking, smoking, and so on. We've gone back after that first pilot, and this is the same kind of picture that what you saw before, but we also have information now. So we did another poll. We also have information about mammography with regard to when did these people have a mammography, and do we see a signature of these cancers, breast cancers, in plasma that may have been missed in mammography. And well, first of all, again, we see a nice separation using both looking at differential methylation for both hypermethylated and hypomethylated, looking at 150 markers or 300 markers. You can see that principal components simply separates out the samples pretty well. And, we're also working on developing tools where we can assign a risk score to a participant as well. And this is where we're at with that. And above that certain risk score, you can see that the risk is going to be lower or higher, and that's what this data is showing. And we also see that these tools don't always work as well for all cancer types in the context of breast cancer as well. Some breast cancers, for some breast cancers, the AUCs are much higher than the other breast cancers. In some cases, and of course, in the context of metastatic breast cancer, this does obviously a lot better than in the context of primary cancer, and that's because there's a lot more DNA shed as well. This is some data for prostate cancer and pancreatic cancer, and just gives you a sense of how well we're doing with these types of tools. All right, so just to summarize, this is where we're at with some of what we've done with CanPath in terms of the cohort. I hope we've convinced you that we can see using some of the material that we've collected, that we can develop new tools for early diagnosis as well. And sorry, this is just a summary of the methylome work. And I just again want to highlight that this is work done by a great team at both in my lab at OICR. And I think that's it for me, John. I think we're going to turn this over to Guillaume now.
Thank you, Philip. That's tremendous. Again, to the group, there will be time for questions at the end, so please put your questions in the Q&A. And Guillaume, over to you. Thank you.
Thanks, John. Thanks, Philip, for the invitation and introduction to talk about CARTaGENE. I will try to describe very briefly what CARTaGENE is, and then I want to present some vignettes, not only from my lab, but from different labs of people that have been analyzing genetic data in CARTaGENE and what we can do with the data available. So very quickly, because most of you are familiar with CARTaGENE, as Philippe mentioned, is the largest Quebec-based biobank. It has a mission, which is to accelerate health record and lower costs of research. It includes 43,000 participants. We just celebrated our 15-year anniversary. And these participants were recruited in 6 regions of Quebec. We have a wealth of information. Some of it is listed here. It includes health questionnaire, diet information, physical measurements. But for today's talk, the focus would be very much on what I put here in the red square, which is the genomic data. So as Philip mentioned, CARTaGENE now has genotyping data for 30,000 participants. We also have whole genome sequence data, high coverage whole genome sequence data for 2,173 participants, RNA-seq data, and whole exome. This data is all available, but I'll focus my description of our results on the genotyping and sequence data. So CARTaGENE has very unique features. I should say unique, maybe not unique within CanPath, but certainly unique worldwide. So we have linkage to administrative health data that can enable very powerful analysis. We have a very broad consent for project both in academia and industry, and there's the possibility to recontact participant. And what is truly unique to CARTaGENE is this genealogical record. And that's due to the very specific history of the population in Quebec. So as you know, indigenous population have lived on the Quebec territory for thousands of years, but that demography has changed quite a bit since the 17th and 18th century due to the arrival of mostly French settlers, such that now about 85% of the Quebec population is so-called French Canadians. And this has obviously implication for any type of studies, including genetic studies done in Quebec. The reason why we are able to reconstruct the genealogy is because of all the very detailed records that were kept in the different parishes in Quebec for marriage certificates, birth certificate, death certificates, so that we can use these records and build an almost complete Quebec genealogy. This is led by our colleagues at the Université du Québec à Chicoutimi, who are involved in the Balzac project. And this is a parallel project to CARTaGENE, but CARTaGENE and Balzac, we work together to facilitate some of these analyses that really can take advantage of both the genetics in CARTaGENE and the genealogy from Balzac. And I will describe some of these analyses in my presentation. So now moving on to the focus of today's presentation, the genetics, I want to very briefly describe what we've learned about the structure of the population in Quebec. It's not only French Canadian, there's diversity in Quebec, and so we've learned a few things by looking at the data, and then focus on clinical genetics and the genetics of complex diseases toward the end of my presentation. This is Quebec, and this is CARTaGENE, this is genetic diversity. On the left-hand side, this is a principal component analysis of 30,000 participants. It is a tool to visualize genetic diversity. It does not represent ethnicity or race or whatnot in Quebec. It's simply a tool, a visual tool to present diversity in the province of Quebec. On the right-hand side, you have a similar tool called UMAP, and you have numbers there. These are different clusters. This analysis, sorry, I don't know if you can see the name here, was done by Alex and Simon Gravel's lab. And for each of these different clusters, at the bottom, what Alex did is label the country from where these participants came from. So for instance, if you look at cluster number 0, most of these participants, which are CARTaGENE participants living in Quebec, have ancestors that came from Haiti. So it's a nice tool, a nice graphical representation to capture diversity and to give us a sense of how much complexity and structure there is in the modern population of Quebec. So what can we learn from such analysis? I'm going to show you a small vignette from something that we learned looking at the Quebec CARTaGENE participant who have grandparents born in Morocco. And this is work by Georgette in Simon Gravel's lab. It's an admixture plot. So for those of you that are not familiar, we're basically asking, can we take the genetic data of these participants and divide it in groups? So when we say K equals 4, we divide it into 4 groups. We have 4 different colors. And I want to draw your attention here to the CARTaGENE participant of Moroccan descent. So you can see that there are 2 major groups. And then when Georgette repeated this analysis with K equal 5, what we observe is that there was the orange group that occur here, that appear in the Moroccan. Georgette realized very quickly that, in fact, when we use the report ethnicity from our participant, from the CARTaGENE participant, this division, between the CARTaGENE participant of Moroccan descent was that in orange you had individual that self-reported Jewish ancestry, and in red, individual that reported Arab ancestry. So, so this is interesting. All these participants, and I'll emphasize, all these participants are Quebec participants, they're CARTaGENE participants from Quebec, and they all have Moroccan ancestry, but they are somewhat different, and this difference can confound analysis, genetic analysis, but other type of analysis. Interestingly, when we look at when these participants immigrated to Quebec, what Georgette realized is that it was consistent with the known wave of immigration from people in Morocco to Quebec, with individuals that self-reported Jewish as their ethnicity moving in Quebec earlier than individual for Arab self-reported ethnicity. And again, this is it is important to know, to have this information, because in analysis, in genetic analysis, this can introduce major biases and confound some conclusions. So knowing about the structure of the participant in CARTaGENE, knowing about the structure of the Quebec population can really help think about strategies to use CARTaGENE to improve public health and health in general in Quebec. There's been also tremendous work by Simon Gravel's lab. Here is an example from Luc, who was a student with Simon, looking specifically at the genetics in French Canadian. Again, on the left-hand side, this is a UMAP of French Canadian, in part from CARTaGENE. And what Luc did is, when you map these participants onto the Quebec map, well, you can see that we can reconstruct in largely the map of Quebec with known subpopulations, strong founder effect, the Saguenay-Lac-Saint-Jean, the Bas-Saint-Laurent, the Beauce. And what was very exciting in Luc and Simon's work is that they could infer from the genetic data and the genealogy data that many of the axes of variation in Quebec were due to the geography of Quebec, and in particular rivers, being the St. Lawrence, the Saguenay, or La Rivière-Chaudière in Beauce. So this work is published, and I invite you to look at it. It's really tremendous in terms of learning more about the genetic history of Quebec. So now moving on to clinical genetics, what can we learn about healthy, or loosely defined healthy population in terms of clinical genetics? And I'll start with one example. They are in the Quebec population, because of its history, so-called founder mutation. This is due to the presence of a mutation that's relatively rare, say in France, but becomes at higher frequency in Quebec just because the settlers that moved from France to Quebec, just by chance, carried that mutation. So one of our colleagues, Dr. Adam Shapiro at McGill, works on this disease called primary ciliary dyskinesia, and approached Simon and myself with this mutation that he had found in several French-Canadian families. It's in a gene called IDEN. And this mutation is very, very, very rare, almost non-existent elsewhere. So what can we do with such observation, with the genetic and in the genealogy? And this is work by Alejandro, again in Simon Gravel's lab. He basically realized that there were 31 CARTaGENE carrier of this very rare mutation. It's a recessive mutation. And then when Alejandro took the genetic data and the genealogy data, he reconstruct, if you want, the ancestral graph of this mutation, and you realize that 22 of the 31 carriers all coalesced very quickly together, indicating that they had a common ancestors. And then, because we have extensive record of who came when in Quebec, Alejandro was able to conclude that that mutation was probably brought in Quebec by two couples that came from a single village in France called La Perche. So type of information that is historically very interesting. I don't have a slide to show it, but the extension of this work by Alejandro is that now we can infer the frequency of this rare mutation in different regions of Quebec, and this can have implication on how we screen for such mutation. In fact, the CARTaGENE data is currently being used to think about improved strategies to screen for rare mutation in different regions of Quebec. as I mentioned earlier, we have whole genome sequence. So it's not on genotyping. We have sequenced 2,100, almost 2,200 CARTaGENE participants. Most of them are self-identified French-Canadian, but we also have individuals with 4 Asian grandparents and 4 Moroccan grandparents. We identify variants, and all this data is available upon request and review. What we did with this data, and this is work led by Ongyu in Daniel Talium's lab at McGill, is we built a browser, allele frequency browser. The simple reason is because it became very apparent very quickly that that type of allele frequency information was very interesting for pretty much anybody. So Ongyu built, for those of you that are familiar with Nomad or Bravo, build a allele frequency server that is very, very similar to that, such that now it's possible to quite quickly query allele frequency, say here taking the IDEN variant, what is the allele frequency of this variant in CARTaGENE, and you have it divided by the major groups that we sequence. It is relevant for mutations that are specific to French Canadian, but just to give you an example, the sickle cell mutation is obviously present in CARTaGENE participant with Asian grandparents. So this browser is, or will be very soon publicly available, and we anticipate that would be a tremendous use for clinical geneticists colleagues as well as the community in general. This is an interesting slide. So I mentioned there are founder mutations that cause disease. I've talked about the IDEN mutation. In the blue diamond here are all known founder variants that are known in the Quebec population. What you can see, as expected for founder variants, their frequency is shifted, so that it's higher in CARTaGENE on the y-axis versus Nomad or European individual in general. So this is nice, but what is even nicer is that if you look at all the black dots, these are all predicted deleterious mutation, and many of them are at higher frequency. So this suggests that CARTaGENE could be useful or will be useful to study mutation that have yet been assigned to rare disease. So there's a wealth of information, even in terms of rare disease genetics, that can be obtained from a population-based cohort like CARTaGENE. We also analyzed the whole genome sequence data to look at structural variants. So on top of the single nucleotide variant, and in DELS we have structural variant. This is work by Rose in my lab. We calibrated these frequency to make sure that our false positive rate was well controlled. And that was the case. Just a few snapshots of what we found that is exciting. The first hypercholesterolemia mutation that was identified in the LDLR genes was found in French Canadian. You have the paper here. This is by Goldstein and Brown, who both won the Nobel Prize for their work on LDL cholesterol. Sure enough, when Rose looked at the data, we did find individuals that carry that French Canadian LDLR mutation. So what you see here is coverage. At the bottom, these are for individual or one participant that is homozygote for the non-mutated allele, and then you can see a lower coverage indicating of this deletion found in one patient. In total, out of 1,800 individuals of French Canadian descent, we found 6 carriers. We could estimate the frequency to 0.14%, and as expected, that deletion, although it's very rare, it is very strongly associated with increased LDL cholesterol. It's not the only structural variant that we found that is pathogenic, and you have here a list of structural variants that are known or very highly suspected to cause disease. Some of these are more frequent in the French Canadian population. All right, so finally, what can we learn about common diseases with a cohort like CARTaGENE? And I will focus on 2 specific topics, genotype imputation and genome-wide association studies very quickly. Daniel Talium at McGill has been interested in building a new imputation panel. I mentioned that the history of the population in Quebec is different than elsewhere in the world, and as such, we might anticipate that an imputation panel that is built using haplotype from French Canadian would impute better genotype in French Canadian. So the first step in building such panel is to phase the data, and the quality of such phasing is assessed through the switch error rate, looking at trio data, I'm going to go quickly through this, but just to say that if you have sequence data from French Canadian, you can impute better French Canadian data. This is as expected, and it's even true for very, very rare allele variants that are seen 2, 3, 4 times in the data set. So with this in hand, Daniel phased the data and built an imputation panel, and then used this imputation panel to impute the CARTaGENE genotyping data. And there are many ways to look at the quality of imputation. I picked that one plot. Basically, what Daniel did here is to compare the number of variants that you can recover using either whole genome sequence data, using CARTaGENE only imputed data, or TopMed imputed data, which would be the go-to right now. So TopMed is a resource where you can easily take your genotyping data and impute it. And so what the plot shows is that most of the variants are recovered using either of these 3 strategies. You also have about 180,000 variants that will be recovered using whole genome sequence and the CARTaGENE imputation panel, but would be missed using the TopMed imputation panel. On the other hand, you will also miss variants if you only use the TopMed imputation panel. But the number of variants that you gain with the CARTaGENE imputation panel is substantial, and it is not surprising, again, given the specific history, the unique history of the population. I don't have a slide to summarize it, but essentially what Daniel decided to do is a meta-imputation where we combine the imputed data from CARTaGENE and TopMed. This data was used to do genome-wide association studies in the form, and this is available through PhiWeb browser. This is work by Vincent Chapdelaine in Daniel's lab. And again, this data will be available very, very, very soon so that you can look up your favorite result, your favorite association online, whether it replicates in CARTaGENE. So time is flying now. I'm going to skip this slide, and just to emphasize what type of new discoveries we can make. This is work by Ken in my lab. We were interested in learning new genetic association with the CARTaGENE data, so we enrich for variants that are well imputed or more frequent in French Canadian. We look at a specific high impact variant or highly likely to be pathogenic non-coding variants. And just to emphasize one result in particular, we found this one variant here with the red circle that associated with TSH levels, so thyroid stimulating hormone. And interestingly, this variant mapped to the TSH receptor gene. It is a variant that has a frequency of 0.14% in CARTaGENE, but it's otherwise very rare elsewhere in the world. It is significant in CARTaGENE, and we could replicate it in Canadian longitudinal study of aging. If you wonder why we replicate it here, it's because there's a fair number of Quebec-based participants in CLSA as well. I will skip this, for the sake of time, we'll skip this example. Just to mention that the data is available and relatively easy to access. CARTaGENE, as the Ontario Health Study, as the rest of CanPath, can make major contribution to public health strategies in Canada. I will skip this. We're easy to find online. I want to show this slide because this is all the people that did the work. And I will acknowledge in particular Simon Gravel, who co-led with me the sequencing project. And then obviously the whole CARTaGENE team who makes all of this possible. And with that, I'll stop here and I'll be happy to take questions.
Thank you so much, Guillaume and Philip before. Thank you for leaving 10 minutes now for questions. We do have a couple that I can start with in the Q&A. And Philip, or sorry, maybe I'll start with Guillaume. You've answered some of these, you've both answered some of these questions already, but just to make it clear for the participants, the audience, one question was, are there any plans for sequencing the samples? If yes, what technology? I think you've both given examples of that. Maybe you could start, Guillaume, because you just you did give some studies using those data, but maybe the technology comment you could.
Yeah. So the 2,200 genomes that we have were sequenced using Illumina short-read. And there's hope to do more sequencing using this technology. there's also lots of hope, but no funding, to do sequencing using long-read technology. And that's something that we are aware and that we want to do, but there's no firm plan right now.
Philip, further comment on that? For example, you mentioned the Genome Canada grant to enable the Saskatchewan and Manitoba project. And for the audience to know, as Guillaume just said, the resources, the data, the resources, the bank, the access for use by the research community is there. But we really do need to work with the whole research community to basically obtain funding for some of this enrichment and analyses that you've started to describe. But Philip, any further comment about the sequencing, the technology, et cetera?
Yeah, no, just okay. So what's been done in Ontario, I didn't really get in to have that much time because we talked a bit about single cell work, but this in the single cell transcriptomics of those participants, we've also done whole genome sequencing, again, using that short read sequencing technology at low pass for some of those participants. And yes, there's hope to get support for doing more. I did mention that in this latest round of Genome Canada funding, one of our regional cohorts has gotten some funding, or some of our regional cohorts have gotten some funding for whole genome sequencing as well. Yeah, I'd actually love to hear from the greater research community that are on this call to hear more about what would they like to see done by CanPath as well. That'd be quite interesting. Many organizations, cohorts around the world have partnered with, as you said, John, the broader research community, including industry, to get stuff out of the freezer and into data as well. So, and I can see that they include companies like Amgen, Decode, and Regeneron. We've been having those types of conversations for years as well. So if there's, again, like I said, I'd love to hear from this audience what they'd love for us to do in that space as well.
Sure. Another question that you've both partially answered, the question, and just to clarify for the audience, then, will CanPath contribute genotypes to large global genetic consortia? I think you both gave examples of how you are doing that in your international projects. And Philip, you mentioned the 300,000 breast cancer collaboration. Those were examples of consortia. But anything else you'd like to add about the what's doable with consortia in international collaboration?
Guillaume, you want to start?
Sure. So
Yeah.
CARTaGENE is already part of several genetic discovery consortia. Myself, I lead effort to understand hematological traits, things like hemoglobin, platelet, and how that relates to disease. CARTaGENE is also involved in anthropometric trait analysis. So these are the two that I know that I co-lead or co-supervise. But I also know that CARTaGENE's data has been used by other labs in Canada, elsewhere in the world, and in disease that I don't work on. So yes, you can contribute CARTaGENE data to international effort, definitely.
Okay.
Yeah, I'll just second that point. That last point that Guillaume made is that anybody can come in and say, I'm interested in using genotyping or some sort of information captured from CanPath to join another consortium. We have, as Guillaume mentioned, ways to access both the samples, the genomics, the data, and so on. It's just a proposal away. And yes, we have been part of these types of collaborations before. One of the things that I was part of was help co-lead was looking at specifically in the clonal hematopoiesis space, how clonal hematopoiesis as a phenomenon impact COVID severe disease severity. And so that was a publication that came out a couple of years ago. And we've been involved as in a number of other GWAS or meta-analysis GWAS type of consortia for various diseases, led either by others or by us as well, using either the Ontario data or smatterings of data from across CanPath.
Yeah. One of the other questions that was here was whether CanPath is open to working with industry. I think you've both answered that. Some of this work is actually tied closely to industry. Anything else you'd like to comment on in that regard?
More and more, we're seeing industry requests for access to the data. And yes, we are certainly having to be aware that that is the future. It's, you know, the world has changed since both Guillaume and yourself and myself we're coming up as scientists. So much more discovery and translation is happening through industry partnership or in industry itself. And so, yes, we're certainly open to that. And as I mentioned before, we're having those types of discussions.
Okay, one question then also, I can start with a partial answer for it, but person has asked why there are no patients with inflammatory bowel disease, and I would clarify that that's actually not true. These platforms are Canadians. These are people living in every province. Some may have inflammatory bowel disease, some may have any illness, but the key thing about a prospective cohort is the ability to follow up over time while their health trajectory reveals itself. That could include people developing IBD. So yes, these platforms are very good for a wide range of investigation. There was one question that maybe that could be clarified. The question was more actually about what kind of basic science could be done around understanding the effects of vaccines. You know, the question was, as vaccines are introduced, are there biological effects? Well, that is actually the world of immunogenetics and vaccinology. However, in the context of large cohorts, Philip, perhaps you with the work you did around COVID, any comment on what type of studies can be or have been done around the progressions following vaccines?
Yeah, I'll just quickly say that, of course, that is something that can be explored. And as well, we could look at how vaccines might be protective or incident protection for other morbidities, John. So that's something we could look at if a vaccine has been introduced to say, prevent one particular condition. It might have a positive effect on other conditions as well. So those are the kinds of things one can also look at, not just about the impact of vaccination or treatment as well. And there has been actually some recent work, not done by us, where, say, treatment for, say, breast cancer has been shown to have a negative impact, or at least an impact on somatic mutation accumulation, clonal hematopoiesis in blood in breast cancer survivors. So those are the kinds of things that can be captured both with clinical and cohorts as well.
Thank you. We're really at the last minute of our time. There are a couple of other questions here, and maybe we can follow up. They're good questions. We respect that, and we'll follow up with each of those points. Yes, there's a lot of work ahead for Canadians to do with a resource like CanPath. And for both of the remaining questions, the opportunity is to work with the research community to obtain the resources to put the data to the best use possible for Canadians. Philip, Guillaume, thank you so much for your presentations today, your leadership in this. Everybody saw the 15-year anniversary. Well, this is a long-term sustained activity that you've both been leading. There are other studies, of course, that can and are being done in related domains. Two very quick announcements that will appear in the chat, but other CanPath activities for people who are interested in knowing more how to access the data. There's something called the Access Hours. The next Access Hour online will be April the 2nd at 2 o'clock. Watch for that. The other thing that's a big opportunity relates to better understanding of the data that we have generated, which can be partly done with the synthetic data set, where there is actually a secure data system developed with funding from Genome Canada and in partnership with LifeBit. That workshop is on June the 24th. We're at time, but again, I applaud the presentations today, the work of all of the researchers and the teams across Canada for making such a resource for Canada possible and now impactful. So with that, thank you all for joining us today. Stay in touch, and we look forward to seeing you again.